Okay, a couple of more groups of platyhelminths. As I mentioned in class, we're not going to spend a lot of time on this class, the monogeneans, just because of time, mostly. These guys are primarily ectoparasites. You might remember that I talked about if you had a freshwater aquarium with fish in it, you may have had a condition called ick. So these guys are tiny little ectoparasites that live on the body surfaces of fish and other organisms that are aquatic. And so pretty much they are adapted to hook onto the outer surface of a host. So you can see these large organs called hapters, and they just consist of complicated hooks and clamps, and you can see the hapter over here and all its little marginal hooks. There's a different kind of a hapter over here. So one of the things about these guys, aside from being almost exclusively ectoparasites, and the fact that they have these posterior attachment organs called hapters, they also have anterior oral suckers. You can see that they have some digestive tract, although it's somewhat reduced compared to turbellarians. And the class name Monogenea implies that they have a single host. They don't have any intermediate hosts, unlike the next class that I'm going to talk about, which are trematodes, that are some of which are also called digeneans. So that implies that there's at least one intermediate host, so a primary or definitive host, and a second intermediate host, hence the name digenean, is one of the groups amongst the trematodes. So pretty much hooking onto fish gills or body walls are where you tend to see these guys and feeding off of them. So that's about all I'm going to say about them. Moving on. Oh, here's, I forgot that I actually had some more pictures of these hapters or epithapters, just showing you the complexity and adaptation of attachment organ amongst these what are called monogenetic flukes, as opposed to the digenetic flukes, which are the trematodes. Okay, so moving on to the class Trematoda. A few generalizations about this class. Again, all parasites. They are structurally more similar to the turbellarians compared to the cestodes. And they do have some digestive tract, although much less reduced, much less diverticulated. So we see an extreme reduction of digestive tract, which falls in line with our parasite adaptation, although the digestive tract still does exist to a certain extent. So in some ways, the cestodes are this extreme parasite, whereas the trematodes, a little less so structurally. But one of the main things that characterizes the trematodes is the fact that they have these really complicated life cycles. They go through all these crazy stages, and excuse me, I'm going to talk about that in just a minute here. So this is the sheep liver fluke, Fasciola hepatica. Of course, that name hepatica tells us something about the liver. And these are huge flukes. You can see in lab, the ones that are on the slides are huge. They're too big to put under a compound scope. You have to put them under a dissecting scope, and of course you can see them pretty well without any kind of a scope at all. So you've got an animal that's at least a couple of inches, again, that does end up feeding on the liver of a vertebrate, and so doing an enormous damage. They do have a mouth along with that digestive tract, and so there's an oral sucker that surrounds that. And then again, you can see a lot of digestive structure in here. This isn't a labeled diagram. We're going to take a look at some other diagrams in a minute. Of course, one of the I'm going to go back here. One of the claims to fame is, or infamous, or infamy is the proper word, is the disease schistosomiasis, which is caused by a trematode in the genus Schistosoma. And there's different species. We're going to talk about Schistosoma mansoni, but there's at least a couple of other species that are really important human parasites in the genus Schistosoma. And again, schistosomiasis is the second most prevalent disease in the world after malaria. So it's a hugely important disease, certainly economically and just from a human suffering point of view. Because schistosomes, like other flukes, require intermediate hosts that are in association with aquatic environments, usually places along rivers where people are living and washing their clothing and using as a sewage place, and you get your water there for drinking and cooking, and so people are using the same water for laundry and for peeing and pooping and eating and drinking, and so obviously health issues arise when there aren't other choices for water treatment. Definitely along the Nile River, one of the most famous places for real problems with schistosomiasis control, and places where there are dams, where water's dammed up and where people just don't have a lot of options. So let's talk about the trematodes in general. Here's the sheep liver fluke. Here is another important fluke. This is not the schistosome. This is Ichthyophthirius, which has a synonym called clunorchis. Sometimes there are just two genera with the same equivalency, so you'll see them labeled both ways in the lab, either Ichthyophthirius or clunorchis. And so it's the same fluke. It's a Chinese liver fluke, is its common name, and it also inhabits this inhabits human livers, where obviously the sheep liver fluke inhabits sheep livers. I don't know if I made that clear earlier. So this Chinese human liver fluke, and obviously because of the fact that it's so prevalent in China and in other parts of Asia is where it gets its name. So you can see the oral sucker here. There's also something called an acetobolum, which is more or less a ventral sucker. There is a pharynx leading to a very simple digestive tube that has only two branches and no diverticulation. So this is intestine right here, coming along the side here and over here. And again, pretty much everything else in here is going to be associated with reproduction. So we have an enormous uterus, we have an anterior testis and a posterior testis, and then seminal vesicles, seminal receptacles. Remember that seminal receptacle is where sperm can be received from a male. Obviously hermaphroditic. They also tend to be simultaneous hermaphrodites, although they are capable of self-fertilization and cross-fertilization, while the cestodes tend to exhibit more self-fertilization. It kind of varies amongst different species of flukes as to the extent of you see self-fertilization versus cross. And again, the availability of another individual is what can determine that. Okay, so take a look at these guys in lab and try to identify some of these different structures here. So talking about life cycles, this is some generalization here, and then we'll look at specific life cycles. Most of the flukes have a more or less called free living stage called a miracidium. And so when you have the egg, the miracidium is a ciliated larvae that hatches out of the egg. Okay, and here you can see it. And then what this larvae does is starts to develop these little balls here called germ balls. Again, the word germ usually means that something is in its beginning. It's starting to grow. It's kind of a generic name for anything starting to create a new thing. And so the miracidium often hatches out into something called a sporocyst, which is a sac that in and of itself produces more of these little germ balls. The sporocysts can then undergo other, make more sporocysts. So you have mother sporocysts making daughter sporocysts. That's not really illustrated on this particular slide, but if I were to draw a circular arrow back to the sporocyst, we say sporocysts making more sporocysts, okay? And then some sporocysts will differentiate into rhedia. Okay, and this slide is just showing 3 different rhedia here. Otherwise, it's all the same thing going on. It's kind of a funny drawing, but the rhedia developing inside the sporocyst, hatching out, and then the rhedia have their own little phase of asexual reproduction where they produce inside their bodies these little structures called cercaria, and then out of the rhedia, the cercaria hatch. And the cercaria are also a free swimming stage, and they have this little tail that kind of they use for swimming around until they're going to find their way into a host. Okay, so each of these 3 rhedia in this diagram are producing 2 cercaria, but obviously they produce many, many more. Okay, so just think about again, the explosive forms of asexual reproduction here. The potential for a single egg producing a single myristidium, which is the ciliated larvae, to produce hundreds or thousands of progeny from there, and they're all genetically identical. So sporocysts producing more sporocysts, sporocysts producing rhedia, many, many rhedia producing many, many cercaria. And again, the cercaria are released into the water and off to find a host. And here's an electron micrograph of a cercaria from, I'm not sure which species this is from. Probably the caption in your textbook says specifically. Okay, so where are the rest of these stages? Well, that depends on the fluke. And it depends on which hosts those flukes have. So as we'll see, there are different intermediate hosts. There can be invertebrates like snails and crabs, vertebrates like fish, and then sheep and humans. So these different stages end up in different hosts. But you get the basic idea behind this reproductive machinery that's going on here. Okay. So these are some life cycles. The Chinese liver fluke or Ichthyophthirius on the top here, and the sheep liver fluke or Fasciola hepatica, on the bottom here. And so these are some examples. I'm not going to ask you guys to memorize all of the different life cycles. If anything, I'd rather have you know the schistosome life cycle, since it's one of the most important. Although these are really important life cycle organisms too. Maybe I would do something on an exam like, say, draw one of the trematode life cycles, and then you can pick which one that you want to learn. So here we have the just starting at the top here, we have the human that ate some raw fish that was infected with the Chinese liver fluke. The encapsulated myristidia, that are inside the eggs, right, are released once they move into the water. Okay, so you have the fluke inside the human body, the eggs that are released, the myristidium hatches out of the eggs. These end up in the water. Notice that there's 2 water stages. There's the myristidium ending up in the water, and then later it's going to be the cercaria in the water. Okay, so first the myristidium are swimming around. They have to find their way into a snail in this case, and this is the intermediate host. The snail is the intermediate host. The fish is a second intermediate host, and then the human is the definitive host. Okay, so the myristidia enters the snail, and this is where it will hatch into a sporocyst and then produce a whole bunch of rhedia. And then when the rhedia burst out to produce cercaria, it's the cercaria that leave the snail body. They actually just go right through the snail body, enter into the water again. Now the cercaria are swimming out in the free environment, and then they have to find their way to a fish, like a carp. Okay, see how complicated this is? And then the cercaria are going to insist in the fish muscle, at which point they're called metacercaria. In lab, you're going to see slides that have cercaria and metacercaria. The cercaria are going to have the little tails. They may have a little fork and they may not. And the metacercaria are going to be like an insisted form, because those are what you find in the fish muscle. Then again, the human eats the raw or uncooked or not so well cooked fish, and then ingests the metasacharia, which hatches out in the stomach. Okay, and then again, the